Question 5 for exam 3. All right, let's get into our question. It's the last one. Everyone wants to go home, and I'm right. Am I right? You're done watching these. You want to be done. All right. Describe the composition of the blood. Include the components of the plasma. Describe the mechanism by which red blood cells are removed from the circulation. Especially explain how heme is broken down and what happens to the various components of the heme. In part 2 of this essay, describe the 3 mechanisms that contribute to hemostasis, i.e. our vasoconstriction, platelet aggregation, and coagulation. Special attention should be given to the role of the platelets and coagulation cascade. For the coagulation process, describe the intrinsic, extrinsic, and common pathways. Also, discuss the role of TPA, plasminogen, and plasmin. All right, let's get into it. For the breakdown of the heme part, I'm really going to be jumping back to the book. It's the best description that I've found so far. I'm going to be quoting from that quite a bit, trying to find the things that I wanted to share. So let's look at our blood composition. Blood is mostly liquid, and it's comprised about 55% of its volume is liquid. Plasma. I read that wrong. Plasma is our liquid component of the blood, and it is 55% of our blood volume. 55% is plasma. Of the plasma, most of that is water, about 90%. Different solutes, proteins, electrolytes, hormones, waste products being transported through the body. All of that is made up into plasma. Our key plasma proteins are albumin, and that maintains our osmotic pressure. Go back to question 4 for that. And it also transports substances, globulins, antibodies, and transport proteins, and also fibrinogen, which is part of our blood clotting. So those are the things that really make up plasma, the proteins that can be found inside of it. Within our red blood, it consists of our red blood cells, our erythrocytes, that's the name for it, white blood cells, leukocytes, there it is, and our platelets, which is our thrombocytes. Releases oxygen into the tissues. Well done. When our cells age and they get damaged, which they do eventually, all cells do, they're removed from circulation by macrophages in the spleen, liver, and bone marrow. As blood transfers through those parts of the body via our capillaries, it is going to be broken down by these macrophages. Heme is our iron-containing component, and that's what really carries the oxygen in our red blood. That is going to be broken down into biliverdin, and that is converted into bilirubin in our tissues. So let's go over that breakdown of heme. Let's focus on that very much. So these macrophages are white blood cells, really. That's breaking down our red blood cells as they get damaged or too old. They'll begin the initial process, and these enzymes inside of these macrophages are going to degrade the chains into amino acid monomers. It's just going to break them down into little tiny pieces, and the heme groups are going to be split into, our heme groups are going to be split into iron atoms, and then we also get our biliverdin. So really it's the white blood cells that are doing all the work with their enzymes inside of them. The iron from our heme is going to bind into a protein called transferrin, and that is going to the transferrin is going to transport it through the bloodstream to the red bone marrow, and it's going to make it re-available for red blood cell production, so that iron is really recycled until it can't be anymore. The biliverdin, again, like I said, is going to be converted into our bilirubin, and that is going to be released into the bloodstream, where it'll bind into more proteins in the liver. We'll get into that. Let's go back to our notes. Sorry. All right, so talking about our bilirubin, it is going to bind to our albumin, and it's going to be transported to the liver, where it's going to be conjugated and excreted into bile. So we're just going to be turning that into something else that our body can use in the bile. In the intestines, it'll undergo another transformation by our flora in our intestines before it goes out of us. Poo-poo. All right, I believe I covered all of that in that section. Mechanism, red blood cells are removed from circulation, heme broken down, various parts. All right, part 2, 3 mechanisms that contribute to hemostasis, like our vasoconstriction, platelet aggregation, coagulation. Let's get into that. Okay, so our hemostasis is the process that our body naturally has to prevent and stop bleeding after vascular damage, vascular injury. Whenever you get a cut, that's our hemostasis coming in there to make a scab. And so our 3 main mechanisms will stop that bleeding. Our vasoconstriction will happen immediately after we get this cut, this injury. And that's going to reduce our blood flow and minimize our blood loss that we have excuse me, because of that injury. So veins are going to constrict real tight. Next step is going to be our platelet aggregation. These are when our platelets are adhering to our exposed collagen at the cut, at the site of the injury. And these are going to release more chemical signals to attract more platelets, leading to further platelet aggregation. Next comes our coagulation cascade. That's really what a scab is. It's coagulated blood. The coagulation cascade is going to be initiated, which will lead to the formation of a fibrin clot. I think they really wanted me to focus on that. Platelets and coagulation cascade. All right, so let's go more into detail with that, with our coagulation. Or first, before we get into our coagulation, let's do more about the platelets. First, there's going to be a vascular spasm, like I said. That's our vasoconstriction to minimize our blood flow to that area. At which point, our von Willebrand factor is going to be secreted by the epithelial cells around it. These will bind to our platelets. So this is our chemical response to this cut that will bring more platelets in to stop this cut. And then our platelets will bind via this von Willebrand factor to our collagen. At which point, alpha granules in the platelets, I'm sorry, I said it wrong earlier. This is the chemical response, our alpha granules. Our alpha granules will release more von Willebrand factor. It will also release more fibrinogen and what's called PDGF, or platelet-derived growth factor. Dense granules in the platelets, so there's a difference between alpha and dense. Dense granules will release adenosine diphosphate, or ADP, calcium, and serotonin. Also, thromboxane will also be released from the membrane, and the receptors of all of these are going to be our von Willebrand receptors and our fibrinogen receptors, which will appear on the surface of any platelet that's activated and pulled into this site. Excuse me while I take a drink. Our thromboxane and our ADP will activate more platelets, pulling them into this cut, into this injury. Gosh. Our fibrinogen will bind to our fibrinogen receptors, which are located on each platelet. And these fibrinogen receptors and the fibrinogen will bind all of these platelets together into one mess. After this, our coagulation cascade will be activated. Geez. So going into our coagulation cascade, it's going to require a couple of proteins that are not super discussed. Well, they're very discussed. It's hard to explain, not as a teacher, not with a giant whiteboard to draw all the stuff out to explain it. But these proteins are called coagulation factors, or clotting factors from the book, and they're normally in our plasma, just sitting there, just hanging out, just waiting. They have different orders for what they can do. The factors are going to be activated, and it's going to lead to the formation of a blood clot once we have our chemical response from this cut. And that chemical activation of the coagulation cascade is going to either be intrinsic within the bloodstream, or it's going to be extrinsic outside of the bloodstream. Either ways, they lead to blood clot formation. I really can't speak anymore. I'm tired. I'm sorry. So looking at our extrinsic pathway. Back to my notes here. Our intrinsic pathway will be within the bloodstream, like I already said, and it's going to be triggered by exposure to collagen at the injury site as we break apart our collagen to bleed, essentially. The extrinsic pathway is going to be triggered by tissue factor, and it's going to triggered by tissue factor released by damaged endothelial cells, and that's going to activate clotting factors in the presence of calcium ions. Both of those will end up with our common pathway, and in our common pathway, it's prothrombin that is converted into thrombin. Thrombin is then going to convert fibrinogen into fibrin threads, and those threads of fibrin are going to be what makes our blood clot. This fibrin mesh will actually trap platelets and red blood cells as it goes along, picking things up after it's been activated, and it's going to form our stable clot to stop all of this bleeding, and that's what gives us our scab. Once this healing goes on, once our clot has been in place, there's going to be a tissue plasminogen activator, or TPA, that is released, and that is going to convert our plasminogen into plasmin, and then the plasmin will degrade into fibrin strands, and that will dissolve the clot over time in a process called fibrinolysis. Fibrinolysis, there it is. Sorry, fibrinolysis. Let me see if there's anything else I need to get into. TPA, plasminogen, plasmin. Got all of that. Extrinsic, intrinsic, and common pathway, fibrin. Dun, 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 dun. I believe that is it. See you later. Have fun.